Hello, everyone. Welcome to our webinar today, where we're going to tell you more about what's been going on in both at Ohio State and also specifically within the Department of Microbiology during these unprecedented times as we learn to adapt and deal with the ongoing COVID-19 pandemic. What we want to show you today is some of the exciting and varied things that are going on both within our department and within the university, and to give you a real sense of the scope of things that our faculty, staff, and students are doing both to respond to this unprecedented crisis within Columbus, within the state of Ohio, within the US, and really across the globe. I hope that you'll be interested and excited to see what we're doing. And most of all, I hope that you'll walk away with a sense of optimism about where we will be when we come out on the other side of this. I know that the more I've learned from this, from some of the people you're going to hear from today, the better and safer I felt as I've gone through this. Like everyone else, I started with a great deal of uncertainty. And as this strange and unprecedented episode has rolled out, I felt more and more comforted and more, to be honest, excited about the future, even though it holds a lot of unknowns. So I won't take up any more of your time. We're going to start with a very good colleague of mine. I've been very fortunate while I've been chair to work with Mike Oglsebee, who's the director of the Infectious Disease Institute at Ohio State. And Mike has, I think, done stellar efforts to bring in so many of us from different disciplines of microbiology and other fields into what I would say is a fantastic example of how Ohio State can work as one university, not a series of small groups, to really face unprecedented challenges in a way that I don't think would normally have been possible in previous times. So I'm going to hand over the mic to Mike now and let him tell you more about what he's been doing.
Thanks, Mike. Let's get the full screen here. I'd like to take an opportunity to provide a bit of context here and describe what the Infectious Disease Institute does and the activities that involve the Department of Microbiology. I start with this, a reminder that the SARS coronavirus 2 was derived from bats. It caused disease outbreaks starting in China, and the disease is known as COVID-19. At the time, I think most people felt that the problem might remain in China. In February of 2020, 98% of the cases were there, 2,200 deaths. But the feeling was that this might move slowly, and that if it does spread beyond the borders of China, we have time to respond. Having trouble advancing here. I'm showing you the picture of the United States at that time in early March. And you can see we have some cases in Seattle, San Francisco, but that's about it. But by February, by the following month in April, you can see it's essentially spread throughout the United States. And this was quite a surprise. It spreads much more efficiently than influenza. And of course, by the present date, getting into June, you can see we've pretty much saturated the country, especially hard-hit areas being in the northeastern seacoast, in places like Detroit and Chicago, Los Angeles, and now you're having flare-ups in Florida and Texas. Now, a lot of people point to the fact that, well, it looks like the average number of daily cases is beginning to decline as a country. And so, yes, we're up to over 2 million cases. We have 116,000 deaths, but should we feel that we're on the descending limb? Well, I share this, which really shows you why the trend is as it is. Much of that flattening of the curve.
Mike?
Yes.
Mike, the slides aren't advancing.
They're not advancing. Let me do this. Oh, shoot. Um, Sorry for this technical difficulty here. And you know what? I think I'm just going to, are we getting this slide? I'll tell you what, I'm just going to talk through this. I'm having some technical difficulties. The point is that this virus has spread throughout the United States, and although we feel like we're seeing a flattening in terms of number of cases nationwide, this really reflects the geography of the spread, because what we're seeing is declining in some areas like the eastern seaboard, but we're seeing flare-ups in cases in places like Texas, Alabama, Louisiana, Florida, Arizona. So we're very much in the midst of the pandemic. We don't have a vaccine in hand, and we only have about 5 to 7% of the population that's been infected. So there's really much more to come. We're in the midst of this. So to tackle the challenge, we really need to muster the resources of the entire university, and that's where the Infectious Disease Institute comes into play. The Institute has over 620 members, 248 faculty, and representative of 14 colleges, of which the College of Arts and Sciences is a significant component, and key contributors to this effort are in the Department of Microbiology. This breadth of expertise allows us to take a very comprehensive approach to the challenge. And I'm going to give 4 major examples. One, we can look at the spillover events and how this virus evolves. How do we go from an animal reservoir like a bat to humans, to a virus that can spread in humans? And so we have efforts underway to look for the prevalence of the virus in the United States in species like bats and white-tailed deer that actually have receptors for the virus. We're looking at domestic animal species like livestock. We're looking at dogs and cats. All of these animal species have coronaviruses. Are they susceptible to the SARS coronavirus too? And is there potential for recombination that can lead to new viruses? We know that cats can become infected by COVID patients. They can shed the virus, but do they contribute to the transmission of the virus in human populations? We're looking at immunity to the virus. We want to look at antibody responses in the human population to understand how the virus is spreading in our populations. We want to know how much of that antibody is a neutralizing antibody, in other words, an antibody that's protective, suggesting that if you've recovered from infection, that you're not susceptible to reinfection. And we're using this knowledge to develop new vaccines. You know that, yes, we're developing, or many people are developing new vaccines, but not all are going to be effective. And so we need to have multiple strategies in play. We're looking at surveillance and modeling in humans to control the threat. Where are the hotspots? What factors are contributing to the problem, and how do we respond? We are helping to inform health departments, city, county, and state, as well as the university, and that's important for reopening the university. We're looking at new diagnostic approaches that may be able to detect viral RNA in saliva. We're addressing bottlenecks in supply chains by developing swabs, for example, for testing. New surveillance strategies that may include detection of viral RNA in wastewater, because we know that viral RNA is shed in feces of infected individuals. And so this gives us a population view of what's going on. And finally, there's treatment. And so treatment could include development of antivirals to directly inhibit the replication of the virus. Can we treat the patient response to the virus? It's actually the cause of disease, the acute respiratory distress syndrome. And finally, develop model systems to understand mechanisms of disease. There's not one solution. The solution is a combination of approaches, and the IDI orchestrates such a combined approach.
All right, thank you, Mike. So, sorry about the technical difficulties we had there, but I think Mike got the point across, and we're going to have a few questions for you later as well. So thank you very much. So the next thing we're going to go into a little bit is, so Mike's talked to you about the big picture of where all of this goes. But there are lots of different sides to this, as Mike touched on. And one of the other things that we've been very interested in the department is looking at more the consumer side of this. So how does COVID relate to our everyday lives, aside from the treatments, the vaccines, the public health, and the other issues? How does it touch on that? And we've been doing a lot of work under the direction of Jeff in the department. And what Jeff's going to tell you about is some of the things that is becoming an increasing interest in how we can look at the virus and how it can move through consumer goods, whether that be cars, whether that be on different surfaces and things like this. And Jeff has got some great things to tell you about that. So I'm going to let Jeff tell you just a taste of the kind of things that he's been leading efforts on in the department. So Jeff, over to you.
Right. So I'm the Center for Applied Microbiology, which includes the Applied Microbiology Service Laboratory. And the goal of the center and the lab is to provide needed services to our local and global community. Opportunity to work on real-world projects while along the way building an industry network. So students fully experience industry projects before going into the workplace, and then they've graduated, they've already established a really strong industry connections. So for the last 2 years, we've been working with our partners in the Ford Research Division on applied microbiology studies. And these focus on controlling bacteria and molds within vehicles with the aim of reducing the spread of bacterial pathogens and to help reduce some of those funky odors that you might smell within your vehicles after your vehicles age, 20 plus years. So research reached out to us, and they were hearing about many of the issues with decontaminating first responder vehicles. And they asked us if we could extend our research immediately to SARS-2. And so Ford Motor Company produces a lot of the first responder vehicles. They have the Interceptor vehicle, and many first responders have really high risk, exposure risk to SARS-2. And so also, if you can at the same time kind of visualize the interior of a vehicle, they're quite complex with many cracks, crevices, and a variety of materials throughout the vehicle. So the challenge is, how does one effectively decontaminate a vehicle? So when Ford heard about the problem, they began exploring solutions, and they thought of the many popular technologies that are out now, such as UV, gases, different types of vapors. But ultimately, they decided to use one of humans' oldest decontamination technologies, and one that's already produced within the Ford vehicle itself, and that's heat. So for context, at the time, we had just closed our labs. We had relocated our offices to our homes. We were 2 weeks into a full shutdown at OSU. And so we were really in the midst of the early days of the pandemic. And I was having car parts shipped to my front door. Ford research team was leaving no stone unturned, trying to locate materials from the Interceptor vehicle. And these were often, they were contacting suppliers who were also shut down as well, working from home. So in the study, we explored the use of SARS-2 virus first. However, the SARS-2, the live virus, is restricted to our BSL-3 facilities, which at the time was reserved for medical testing. And it's also very costly and high risk to operate in. So we looked for a safe virus to work with that was physically very similar to SARS-2. And from recommendations from our colleagues, we began using a safe coronavirus that's not pathogenic to humans. And in the study, we tried to mimic the conditions within the vehicle. It's an application-based study. So we took defined conditions from the background work that the Ford team had completed, and we took fabrics, plastics, vinyls that were used to produce the interceptor vehicle. And we studied a temperature that could be produced within the vehicle and was shown was very likely to neutralize the virus, which was about 56 Celsius. So in the study, we found that the surrogate virus was neutralized on the vehicle materials after the surface had reached 56 Celsius and was held at 56 Celsius for over 15 minutes. So then Ford took our data, they used it to design this really elegant solution. So now if a first responder is transporting somebody with COVID and the virus is left on a seat or a handle, they now have this other option to be able to disinfect their vehicle, and especially for all those high touch surfaces that are quite complex that are really difficult to reach if you're using a standard liquid disinfectant. So there are many routes to the spread of SARS-2 virus, and we've received a ton of interest in validating the effectiveness of decontamination technologies from many different companies. And as we continue to live with this virus, it's important to have this service available for manufacturers, for users of these decontamination technologies. So finally, I just want to thank Ford research team, my partner in this study, Jesse Quick, and all the support that we received from the Center for Applied Microbiology team and from the Department of Microbiology and the Infectious Disease Institute.
All right, thanks, Jeff. So as I say, this is just a kind of a flavor of the kind of thing that Jeff has been doing with the Center and the Applied Microbiology Service Lab. Some of you may have got the notice that when we originally planned this meeting, we would be talking about our services to local microbreweries in Central Ohio. Don't worry, we haven't forgotten about that. It's still going to happen, but this is really taken over right now. So it's been an interesting time, and I want to thank Jeff for the amount of effort he's put into this. It's been quite stellar. It's really been quite remarkable. And one last thing we forgot to the next speaker, this is where we've also worked with the Infectious Disease Institute to great effect to really maximize our ability to, I think, to make a big difference in this area. So switching gears a little bit, so that's more of our kind of applied microbiology side. In our department, we have a huge amount of basic research. I have some world-class experts you're going to hear from next, a couple of the faculty who've been working on different aspects of the COVID-19 pandemic. And what we've tried to do here is to give you a little flavor of the different things we do. So we're going to start off with Irina, who's someone who's been at Ohio State as long as I have. We came in the same time back in 2001, and she's a world leader in a study of a group of proteins called RNA polymerases. And to those of you who remember when you were a microbiology major, you may remember that this class of proteins has been a very effective drug target in the past, both for HIV and for tuberculosis and for other infectious diseases. And so what she's been looking into a little bit is how viable this might be in the future as a target for treating COVID-19. So I'm going to hand it over to her now, and she can tell you a little bit more about what she's doing in the lab right now.
Do we see my screen? So if we,
yes.
Good, thank you. So as Mike said, I have been working on what you have been reading in textbooks for most of my professional life. So the central dogma of biology is that all cells make RNA out of DNA using RNA polymerases, and then ribosomes make proteins. But RNA viruses are very different. And of course, RNA viruses have a lot more than just COVID. You have influenza, hepatitis, Ebola, polio, and so on and so forth. So all these viruses, instead of DNA-dependent polymerase, use what is called RDRP, or RNA-dependent RNA polymerases. Cellular life forms such as us or bacteria don't have these enzymes. and that makes them attractive targets for selecting antivirals. As I said, this is a proven drug target. These enzymes are highly conserved at sequence and structures, and we have more than 25 drugs FDA approved for treatment against influenza, hepatitis, polio, and so on. So remdesivir, which people heard about a lot, I'm sure, is one of these. And it looks like a nucleotide that RNA polymerase normally takes and incorporates into the RNA. So the way these drugs work, so if you have normal RNA synthesis, RNA polymerase selects a nucleotide according to the template, adds it to the chain, and does it many times. In the case of polioviruses, 30,000 times. When it incorporates remdesivir, the chain stops and no further nucleotides can be added. So now the problem is that coronaviruses are actually very different from all other RNA viruses because they uniquely encode an enzyme called exonuclease, which recognizes drugs as remdesivir as errors during replication, excises them, and allows the RNA synthesis to restart. So this is why, in part, but not completely, the remdesivir is not a perfect drug. And it was, of course, selected as an inhibitor of Ebola. So when I started paying attention to this topic, I realized that also, unlike other viruses, coronaviral RDRP has 3 proteins in it, and only one of them is similar to others. And furthermore, that there are at least 9 proteins that come together. And as this complex, they mediate all steps in viral replication. And that gives us multiple targets for drug discovery, because in addition to just nucleotide polymerization step, you can inhibit more. And what I also realized and made my lab completely switch to this topic is that COVID-19 is obviously a pandemic, and it's already the 3rd case of deadly coronavirus in our century. MERS and SARS were previous ones. And as Mike Oglsebee told you, we have a lot of zoonotic viruses. We have them in bats and in other hosts, and they are all potential threats to us. And coronaviruses are the largest known RNA viruses, and they are very complicated. And what is good from my perspective about them, that this replication transcription complex that I'm talking about is highly conserved. So in this figure, things that are red are the high conservation score, more than 80%, and all the proteins that compose the replication transcription complex are red, and the surface proteins, which, for example, commonly used as vaccine targets, are considerably less conserved. So the plan of mine and my lab, and we are already working in the lab, is to use a combination of biochemistry to identify drugs, potential drug candidates using in silico drug discovery, and also library screening of compounds, and use structural biochemistry in collaboration with our colleagues in Germany to basically try to find drugs and save everyone. So that's what I am doing. And I am trying to stop the screen share.
All right, well, thank you, Irina. So as you can hear, this is exciting and ongoing, and something that I think a lot of people are trying to do. And I'm glad that Irina's on our side in this, because she's very, very good at this. So thank you. The final speaker before we get into the Q&A is Matt Anderson, who's an assistant professor in the department, who joined us a few years ago. And this was a hire we were very excited to make together with the Infectious Disease Institute. Matt works in a range of different areas. He primarily works on fungal pathogens, but as you'll hear, he has a lot of other research interests around infectious diseases and how they can target and spread differently through different populations. And specifically, he's going to tell you about indigenous American populations and the very challenging public health concerns there. But I would say that Matt is really quite a visionary young scientist who looks at a lot of different ways of coming at some very, very interesting problems. And so I warn you, Matt, I've got quite a few questions from the audience already that I'm going to be shooting your way afterwards. So you're all going to hear a lot of very interesting things from Matt. So I'll hand this over to Matt now, who's going to tell you about some recent work he's been doing about COVID-19 and public health concerns in particular populations. Okay, over to you, Matt.
Great. Thanks, Mike. So, Sioux Nagada, hi everyone, I'm Matt Anderson. I just want to acknowledge the fact that right now we're on land that historically has been Miami, Shawnee, Wyandotte, and Potawatomi land. So within the work you've heard about COVID so far and more broadly in media, what you've probably heard more than anything else is the fact that racial disparities exist within different populations revolving around COVID-19. One second. So when we're thinking about the impact of the virus on different folks within the United States, each community needs to be kind of contextualized within the framework in which it exists in the United States, as well as in their state and in their local environment. So what's been reported repeatedly is the fact that there's health disparities in the incidence of COVID-19 infections, as well as the outcomes of death across the US. You can see here that illustrated by the color coding, where more intense orange colors demonstrate a higher burden of both cases and deaths for different African American populations within the United States. This is true not just for African Americans, but it's also been found true for different Hispanic and Latinx individuals. And where data exists, as reported in this Journal of the American Medical Association article for American Indian, Alaska Native, and Pacific Islanders, so indigenous people within the United States. So thinking about specifically that last part, where data exists for American Indian, Alaska Native, and Pacific Islander populations, that needs a little bit of context to be able to play out or fully understand. So within the United States, specifically showing here the continental United States, there are 574 federally recognized tribes and 325 different reservations, so physical land masses that have been allotted to those tribes in payment for different land bases that were given up or other treaty obligations. Altogether, there's roughly 5 million people within this Native American, Alaska Native, Pacific, specifically Native Hawaiian category. These people have very specific relationships within the United States that sets them apart from every other population within the US, specifically the fact that the United States recognizes tribes as domestic dependent nations. So what does that mean specifically? What that means is that each tribe within the United States that is federal recognition has the authority to establish and enforce their own laws. So they have governance above and beyond what the United States and the state and local levels have. Tribes also have a right to establish and define a relationship with different entities. These can be individuals or they can be different nation states or tribes. Also, there's this relationship between the US and tribes that has been codified into law through treaties that require different obligations from the United States through treaty rights, access to different land bases for fishing, ceremony, access to different provisions that were set into that law during the establishment of reservation land masses, etc. So that means that the interaction between the federal government and different tribes is somewhat unique within the context of the United States. We're thinking about that in the COVID response. We can see that tribal nations have been very hit hard by this epidemic. I'd like to start by kind of highlighting the Navajo Nation, which is located here in the Four Corners region of the Southwest. The Navajo Nation has a landmass of about the size of the state of West Virginia. And at one point, not too recently, they had the third highest rates of COVID-19 infection, only behind New York and New Jersey. They were hit very soon in the COVID-19 epidemic, and the death rate has kind of continued to climb at a steady pace. So their experience with COVID-19 is in no way either reduced or changed since the beginning. Within the Navajo Nation itself, there are a number of different counties. These counties can be parsed into two, either Navajo counties or non-Navajo counties, based on whether or not Navajo people are kind of the dominant population within that reservation county. What you can see is that even within the Navajo Nation itself, there's this disparity between the incidence of COVID-19 infections in Navajo counties that are primarily Navajo Nation citizens and non-Navajo counties that are going to be a mix of both Navajo citizens and non-Navajos on Navajo land. So there's this very complex relationship in COVID-19 infection and tribal communities. A lot of this probably revolves around this idea called social vulnerability. So what is plotted here is the social vulnerability index, this measure of the resilience of a community to some kind of external stress that's placed on it, specifically around human health in this case. And the social vulnerability index of Navajo Nation, shown in blue on the extreme left, is higher than you're going to see either in 3 states that include the Navajo Nation area, Arizona, New Mexico, and Utah, or New York City or Seattle, 2 places that have been hit fairly hard. And the reasons for this social vulnerability being so high in Navajo Nation include a number of different disparities that exist with regards to basic features and clinical care. So a large proportion of the Navajo Nation does not have access to running water or electricity. There are only 13 grocery stores on Navajo Nation, which is about 1/10 the number that exists within the state of West Virginia. Only 6 hospitals and 7 clinics treating these folks, and there's a number of pre-existing health conditions that kind of exacerbate the issues, all of these issues simultaneously. To give you another kind of depiction of the importance of COVID-19 within Indian country broadly defined, I'd like to highlight another reservation, the Cheyenne River Reservation, which houses the Cheyenne River Sioux tribe, located here in South Dakota. This has been a partner community of mine and some others that I've been working with for quite a while now. Houses about 11,000 people within an area the size of the state of Connecticut. It faces a number of the same issues that I mentioned with Navajo Nation. And Cheyenne River has been very active in its response to COVID-19, specifically by shutting down its borders in something that's actually made national press. So there are checkpoints at all major areas getting into Cheyenne River Reservation, where they have the right, based on some of those obligations that I mentioned previously, that define that domestic dependent nation status, to be able to turn people away from their borders. Within this COVID-19 response, they were also very interested in knowing what or whether or not there are infections within their population already. So we have an ongoing project that's about 5 years old now with the Cheyenne River Reservation. That's a larger study of autoimmunity within Cheyenne River Lakota people. And specifically within the context of this study, I've been working on microbiomes. So how do the gut microbes associate with rheumatoid arthritis within this population, because they have a much higher incidence of it than the general US. So we've collected fecal samples throughout the beginning of the year up through March from participants as part of this study. And the tribe reached out to me specifically asking whether or not I could look through these different samples to identify RNA indicating an active COVID-19 infection in any of the participants. At this point, we had 30 samples and had the capacity to be able to run them, whereas the Cheyenne River Reservation itself only had 22 kits to be able to test COVID-19. So our capacity here in the Department of Microbiology was larger than the Cheyenne River Reservation itself, which again goes back to those treaty obligations the US should be fulfilling to that nation itself. Luckily, we didn't find any identified cases within this population, but at the same time, it's allowed us to be able to begin building out different abilities on the Cheyenne River Reservation itself that lead to empowerment, be able to begin processing their own samples in-house and store and analyze their own data. Thank you.
All right, great. Thank you so much, Matt. That was interesting. And I think it gives us a kind of a great insight into things which are happening more generally, and really shows us how the challenges are unique and different to what we're used to seeing. So what we're going to do now is to go through a little Q&A session. So thank you very much. A lot of you have sent questions in. So I'm going to start by going through some of the questions that were sent in and direct one to each of the panelists. And then we're going to have some of the live questions that you've been sending in as the webinar's been progressing. So I'm going to start with a question for Mike Oglsebee, because I know that he has thoughts about this question and he's been asked it a lot. And this is because Mike's been on some task force at Ohio State and in Central Ohio. So Mike, can you tell us about the science behind the mask, no mask policy? What should we be wearing masks? Should we not be wearing masks? What's the general, in a nutshell, what's the take-home message on masks?
I'll give you the answer first and then the rationale second. Masks are effective at preventing somebody who is shedding the virus from infecting others. So this is a respiratory pathogen. It replicates initially in the nasal cavity and pharynx. And so as you breathe, cough, speak loudly, you're releasing droplets, which can be inhaled by others and initiate an infection, or it can land on a surface where somebody touches that surface, touches their face, and the cycle's complete. So what the mask does is it prevents those respiratory droplets from spreading in the environment if you're shedding. What masks do not do, or at least we don't have evidence of, that they are not a significant source of protection for somebody acquiring an infection, potentially acquiring an infection, at least with respect to a surgical mask or a cloth mask. There are respirators, like N95 respirators, that can protect against inhalation exposure, but that's something that we want to restrict to healthcare workers that can't physically distance themselves from infected patients.
All right, so thank you. So masks are a good preventative measure because you may not know when you have COVID-19. Okay.
Particularly since, just to make the case, we know that you shed significant amounts of virus up to 2 days before you're symptomatic. And so that's key.
Yeah. I think there's a huge body of evidence about the general effect on restricting the spread with masks. Jeff, a question for you, because this has come and gone a lot in the media. It was a lot of concern at first. So where do we now stand about viruses on surfaces? How long do they last on surfaces? When should we and when shouldn't we be concerned about COVID-19 infections via surfaces? Just a kind of snapshot answer.
It's kind of been really interesting to explore this, because there's data that covers room temperature to about a high temperature of 95 for surfaces and a few different surface types. And you might see it really depends on humidity and time. So at room temperature, you might see it for several days. It's really just dependent on the surface type as well. We're really interested in exploring the area between 95 Fahrenheit and about 120 Fahrenheit, because that's kind of the area that's been unexplored as far as survival on surfaces. So I think it's in terms of looking at the surfaces that you might also have in your house, it's been pretty well established about survival of coronavirus on shipping and packaging materials. It's very short, so those people shouldn't be too concerned about.
All right, that's very reassuring. Irina, I got a two-part question for you. So you can probably guess what this is coming at, because there's been a lot in the news recently about this. So if we think about drugs rather than vaccines, so how much scope is there to target novel virulence factors in COVID-19? And do you have any thoughts about these very recent studies that came out about dexamethasone and how it seems to be a very, very effective drug, unexpectedly effective? So what do you think the drug route is, given that for some viruses, we've never ever had effective vaccines?
Well, there are obviously two routes for drugs. So there are anti-inflammatory drugs. and I think they definitely work. Unfortunately, we have a lot of them that they are very cheap. But I think, well, and Mike Oglsebee can correct me if I'm wrong, that the inflammatory response in us will differ a lot depending on which virus we are infected. The COVID appears to be particularly good in inducing cytokine storm. So, and of course, the other part are antivirals. And people have been trying to do antiviral discovery for a while. And essentially, that's what we are trying to do, because we simply are trying to find drugs that are likely to target a wide variety of them. And we, as I said, I just never realized that we don't know enough about the system. But these are two different classes of drugs. And I think, as in everything else, a lot of drugs will be tested and many of them will fail for a variety of reasons, a lot of them for toxicity or pharmacokinetics. And we just need to put we have to have a lot of baskets of as many eggs as we can have and to try it all at the same time. Something will work, but there is no guessing of what it will be.
Okay, thank you. Right, so I've got a question for you, Matt, which I think is something that has come and gone, but I think is coming back more and more. So you mentioned with the microbiome. So do you think that this is an opportunity really for probiotics to help support a strong immune system that will improve outcomes for people, or just generally that's something that we should be thinking about more, given that there seems to be very different outcomes depending on how robust people's immune systems are?
That's a fair question. So I would be careful about highlighting any potential benefit from probiotics here. Probiotics seem to be still fairly hit or miss, and their track record has not been great. The infection that you typically get is going to start in the respiratory tract, which probiotics are less likely to really have as much of an impact on. And that's where the cytokine storm initiates and kind of builds outwards. And it's turnover of different microbes in the respiratory tract that is probably leading to the cytokine storm. This is true in other viral infections, but hasn't been shown in COVID yet.
Okay, thanks. That also addressed someone's question a little indirectly about the cytokine storm. So I'll follow up. We did have one question that I think Mike can confirm here, that this is a case of being very careful what you hear. Someone said, lately we heard about asymptomatic people not being able to transmit infections. Is that true? No, that's not true. Asymptomatic people are very, very good at transmitting infections. And if any of you took microbiology at Ohio State and remember, the most famous case of all is Typhoid Mary, who repeatedly transmitted typhoid without ever displaying symptoms to literally thousands of people. So this is something to be very, very careful about. So I'll throw out something that I don't know who wants to come at this, because this is a very interesting question that I've had as well. We hear about a lot of different vaccines at a lot of different stages. So what kind of criteria will be used to decide how to move forward with the vaccine? When we hear that there are tens of potential vaccines coming through, what should we expect from this? Is this normal? Is this abnormal right now? Should we be optimistic? Mike, do you want to take a stab at this first?
Yeah, and keep in mind that we're taking multiple very different approaches to vaccination. Some are RNA only, which is probably on a safety standpoint, it's really great, but you're asking a lot for the immune system to respond in a robust manner, because that RNA has to penetrate cells, produce proteins, and then you have to have a robust immune response. You have the viral vectored vaccines, which gives more of a boost to innate immunity. And some of these vectors don't replicate, so they're a weaker stimulus. Some do replicate, they're a stronger stimulus. And I will tell you that those that are in the pipeline kind of follow that sequence. So the Moderna vaccine is an RNA vaccine. It's probably going to be very safe. It's a high risk in terms of efficacy. And then as you get into the viral vectored vaccines, they're a little farther in development, but they will give a more robust immune response. all of these companies are already ramping up production so that in the event that the phase 1 and phase 2 trials turn out well, they will be ready to distribute a product. But again, it's reasonable to assume that based on animal studies, some of these will be effective. It's just, are we going to have some that are more effective and provide the level of effectiveness that we want? So I think it makes a lot of sense to have multiple products in the pipeline. Does that?
Okay, great. Yep, I think that comes at it. I think it's fairly standard. If we look at, my take on this is we look at the way that in an analogous case with HIV, where there is no vaccine and there is no one silver bullet to cure HIV, but there are lots of ways to treat HIV using various therapies.
And Mike, could I add one piece to that? Because I saw this in some of the questions that came up earlier, and it was questioning, why do we think we can make this happen with coronavirus? And I think it's important to note that, this is an acute virus infection. You do seroconvert, you do develop an immune response that clears the virus. That's very different than HIV. And we've also been successful at developing vaccines for animal coronaviruses. And so there's every reason to believe that we can make this happen here.
So this gets into another question, just a quick answer. So we're hearing a lot about antibody treatments, so human plasma surrogates and things like this. Where do you feel that's going? Anybody? Mike or anyone else? Mike?
Yeah, I think the immunotherapy is going to be quite effective, but that's going to be in individuals that are suffering very high viral burdens. Those individuals also happen to have very significant cytokine storms. And so I think you would either use it in those that are suffering a severe outcome or those that are at risk for a severe outcome that are in the phase where they still have significant viral burdens on board. It makes sense. And we know that we can identify neutralizing antibodies.
But we also have humanized cows, and those can actually produce high titers, and they don't have, I think, cytokine storm. So maybe this will be best. And they are big,
I've got a very interesting question that came in. I'd like to get Matt's take on, because I know that Matt has experienced some of this and knows that this is an underappreciated issue. So someone asked a very interesting question about how, when we have this kind of all hands on deck approach to a pandemic, that there may be some dangers as to what happens to people's data. And specifically, the questioner asked whether this way that there's a switch between federal funds and private funds and data sharing mandates, do you feel there's a risk for data theft and biopiracy that would target people of color and indigenous people during this time? Because that has been a problem in the past. Do you have any thoughts about that?
Right. It's a good point. It's likely happening. And as soon as an epidemic occurs, a lot of the regulatory steps that exist seem to fade a little bit to speed up the process of science. So yeah, that's going to be a real issue. And I think the only way to combat it is by continuing to raise instances in which it happens and educate researchers that this is something they need to be very thoughtful about and conscientious about when they're negotiating with different entities.
I think that's a very thoughtful answer. And I think I agree with you. People tend to underappreciate this, and just, it's a historical problem which has happened again and again in East and in other countries. So that's an interesting point. Okay, Jeff, people had wanted some, when you said the virus doesn't last too long on surfaces, how long is not too long and how long is too long? I know it's how long is a piece of string, but Jeff, you kind of put yourself out there on this one.
It's true. So I'll speak to kind of what we have observed in the lab. There are a lot of studies right now that talk about room temperature survival, and room temperature survival on packaging materials, cardboards, but also on stainless steel and plastics. And room temperature, that can vary pretty widely, from 24 hours we've seen on cardboard to, I think I've read as long as, I thought it was 14 or 16 days on some surfaces like stainless steel or glass. And so what we've seen in the lab is that we try to target within 24 hours. When we dry our viruses on the surfaces that we use, we target the use of those within the test within 24 hours, because we see a large percentage of the virus die off just in that initial dry time. And the coronaviruses are usually the most susceptible viruses to disinfection, to any types of treatments. And so they're pretty, and think of all the different types of viruses, they're pretty on the weak end of viruses, so they can pretty easily be disinfected. And so we see a large percent of it die off just by drying at room temperature over the course of 24 hours.
All right. Yeah, so there's a lot of different follow-up questions, so I hope this doesn't seem too random to everybody. So I think this is for you, Mike, because I know you've been in the middle of this. Do asymptomatic people clearly test positive, or is there a kind of continuum here?
There is a continuum, and so you can imagine somebody's been infected. If they're going to develop symptoms, there could be up to 14 days. So you could imagine you might test viral negative for several days following exposure, and perhaps only test positive as you approach this phase where you may become symptomatic. If you're destined to be asymptomatic throughout, the level of virus is going to be lower throughout, and so you could test negative, positive on a third sample. So it's very much time dependent and very much dependent on the viral load. There's about a 20% false negative rate overall for viral RNA testing based on nasopharyngeal swabs.
Okay, so I got another question for you from someone we both know, whose name I won't disclose. Anyway, so it's an interesting question because I know we've discussed this and I don't know what the answer is, but I know it's more of a public health question. So early on during the pandemic, there was really no recommendations for mask wearing early on, say at the beginning of March. Was that because of a supply and demand issue, or a need to reserve for medical healthcare professionals, or really a lack of appreciation of the efficacy in general of wearing masks? Why do you think we've come a little late to the mask issue?
It's a great question. It's actually all of those points that you've raised. The evidence at a population level of the efficacy of masks was quite limited. And right now it looks like masks are effective, but when used in combination with distancing measures, good hand hygiene, masks alone are not the solution. So that's part of it. And then there's also the concern about if you don't know how to properly don and doff a mask, you could actually enhance your potential for inoculating yourself. In other words, increased touching of your face, your eyes, your nose. In the process of taking it on and off, you could enhance the potential for infection. And yes, there's also the concern about people, at least initially, rushing for N95 respirators that are very much in need of, and we're in limited supply for the healthcare community.
Okay. So someone had a very good question here that I'll answer because I've been very interested in this as a kind of more of a basic scientist. But so someone asked, we're hearing that more testing means more positives. The general public doesn't understand the error here. How would you explain this? So I think this is very simple. The total number of positives is not really the issue here. It's the percentage of positive tests in the total. And what CDC guidelines discuss is not your total numbers or anything else. It's the percentage of positives. So I think Mike can correct me if I'm wrong. The decision to open up a little more in Ohio were based upon the total positives dropping around 5% or less. And the ideal number when there's good control will be around 2 to 3% of the total tests being positive. The reason that more tests are important is because it gives you more confidence in the strength of that percentage value that you're looking at. So I think people have got to start to understand the need here to look at the numbers as the percentage total positives. And we like to look at the number of new cases because it's easy to look at, particularly the number of deaths and the number of ICU admissions. But if you look at the percentage of positives and the percentage capacity of ICU units, these are the real metrics. And these are the numbers that are being looked at in the states that we're hearing are potentially problematic right now. So I think it's looking at this, not as tools, but as percentages to see what they mean. Let me see. So I'll ask you, I think I'm going to ask you all for sort of a wrap up here because we've only got 4 minutes left. So I'll start with you, Mike. What's your most positive takeaway now on the pandemic? You've been very deeply immersed in all of this. What's your positive takeaway?
Well, I like looking at numbers in new cases in Ohio, in New Jersey, New York, in the Detroit area, because it says that by modifying viral behavior, we can definitely at least get this level of population infection to a level where we can contain it. Particularly if we continue monitoring and we have contact tracing methods to give us time to develop a vaccine. I find that very encouraging.
Okay, that's great. Jeff, question for you. So you've been doing a lot of work with this around COVID in the kind of inanimate part of this with consumer goods and other things. Do you worry about, or do you think we should not worry about acquiring COVID through inanimate objects? Very simple question. What's your takeaway on this from what you've learned in the last 3 months?
I think our initial tests, when we were looking at dry times, I was very encouraged by that. Seeing multi-log reductions from an untreated virus on a surface that over just 24-hour period, we'll see that multi-log reduction. I think that's very encouraging. So I was one of those people that was bringing groceries home, unloading them, spraying them with ethanol, letting them sit in quarantine for a while. And I think after seeing those dry times and the reductions, now I feel much more comfortable with not worrying so much about material and surface transmission as much.
Okay, that's great. Irina, how confident do you think that we'll, from the drug treatment and the antibody side, the anti-polymerase treatments, how confident do you think that this disease can be controlled?
I am actually pretty confident. I think we will have both drugs and vaccines, hopefully more than one. The question is more to the point, is that what we failed to recognize that this pandemic was coming. And so I'm seriously concerned that with air travel and everything, we will have more. And I think we just should be doing more forward thinking and anticipating what could be next. But there are very serious efforts all around the world. Like in Berlin, for example, they're dedicating an entire institute to identify various drugs against RNA viruses, essentially. So I'm sure if there's many people working, we will find something.
All right. The last question I'll leave for you, Matt. The pandemic has intersected with social justice issues and other things, and in fact, it's highlighted how deep-rooted some social injustice issues are. Do you feel that there's an opportunity here to improve both public health awareness and public health outcomes in what essentially are underserved and in some cases more vulnerable populations? Do you feel that there's more that can be done here? Do you see a route forward through this?
So there's always certainly more that can be done, and there's certainly always the opportunity to make that happen. The question becomes fundamentally about public will. At what point does it become a flash in the pan and people forget versus something that they really push for institutionalized changes to occur? And until that happens, it'll just repeat itself. I think there's momentum here. Things could change, but we'll have to wait and see.
Okay, so think we're done here. I think that we've just hit exactly 1 o'clock, so I appreciate that. Thank everyone for their time. So it just remains for me to thank the audience. Thanks so many of you for turning up today and hearing more about what we're doing. I'm sorry we couldn't get to more of your questions. A lot of great questions came in. We just didn't have enough time to touch on all of them. If you want to follow up with any of us, please, our details are on the website. You can reach out through ASC as well, the people who invited you to this, and they can direct you to us. And I hope you've come away with a sense that we shouldn't be pessimistic right now. We're living in challenging times, but I hope you can see we at Ohio State in microbiology and many others are making sure that we'll come out of this, I think, with an appreciation of what the challenges are, but be much better equipped in the future to go to face these things head on. So thank you again, and I wish you all a safe week.